Or you can think about
it's
it
fine.
too.
Yeah.
Okay.
Yeah.
Okay. Yeah. It's it turns out it's easy to sort of crop things, take things out, put them down,
Right.
uncrop them later. So we have lots of flexibility.
Sounds good.
Okay, let me just. Oops. Okay, looks like we are all set on that. It's 11, so we'll just give people like another 1 minute to finish joining up. So thank you both so much for agreeing to speak today. I'm really excited about both of your talks. I know it's a little bit hard to like cram it into the shorter time, but sometimes it's fun to do the, highlighting multiple people option. So
yeah, definitely. Thanks for the invite.
Yeah, absolutely. Jude, so do you know Madeline Uden personally, or she recommended your work to me, and so you just started, right? Like you're a very new professor.
Yes, I started the end of September last year. Yeah.
Between two of us, I'm senior by a month.
By a month.
Oh, okay. Well, I thought you were a little older, Shashank.
I am a tiny bit older than Jude as a professor. Yes, absolutely.
Academically older. Okay, well, that's perfect. Usually we try to have the less senior person go first, but of course, there's not a huge difference between the two of you. All right, perfect. So maybe just in the interest of time, I'll go ahead and get started, and people always continue to sort of filter in. So good morning or afternoon or evening to everyone here. Thank you all so much for being here. Today, we're delighted to have two early career researchers, Jude Phillip and Shashank Shekar. And so we're just sort of evenly dividing the time up between the two of them. So they'll each have like 20, 25 minutes of presentation, and then we'll do questions, sort of the usual format. And so I think everybody knows what to do by now, but just raise your hand for the question function, or type it in the chat, and we'll do all the questions at the end. So without further ado, whoops, did I just mute myself? No, okay. Still getting used to Zoom after all these years. So without further ado, I'm going to just introduce our first speaker. We have Jude Phillip. He is a new assistant professor of biomedical engineering. He also has appointments in chemical and biomolecular engineering, oncology, and he's also a core member at the Institute for Nanobiotechnology, and he's at Johns Hopkins University. His lab studies biological aging dynamics in the context of health and disease, and he combines fundamental engineering approaches with translational aging and oncology research to develop strategies and technologies to probe aging and disease with the goal of identifying mechanisms to modify aging trajectories, excuse me, to drive health. So that sounds very important, and really excited to hear about it, and sort of a different take than some of what we've been talking about so far at cell migration seminars. So really excited to have you, Jude, and please take it away.
Yeah, thank you so much. And thanks so much for the organizers and for the invitation to speak and present some of our work today. So I'm actually really excited today to share this work with this community. I think some of the work we've been doing over the years really fits in well with some of the talks that we've heard so far, and I've really enjoyed listening and learning through all of them, actually. So I chose to speak on sort of like a broader topic, these emergent patterns of cell migration in health and disease, particularly because I'll give a little bit of flavor of some of our work with aging, and then the ways that we're extending this technologies and frameworks to really address multiple other diseases and other health aspects. So my main motivation and one of the key goals of my lab is really to understand how dynamics of aging and the ability to use these measurables, whether it be at the cellular level or the clinical level or the molecular level, really to understand how we age and how we can impact people as they age. So over the years, quite a body of work have been growing in the sense of what are some of these features that actually change as a function of age? And there's been this really nice paper published back in 2013 that really goes and shows some of these hallmark features of aging. And here we can see aspects of telomere attrition, we have epigenetic alterations, and a lot of these have focused on more molecular-based changes, right? So how these changes in molecular networks really impact aging. And, being an engineer myself and, thinking a lot about cell biophysics, I felt that there's a lot of ways that cell biophysics could really fit into this whole paradigm of aging, right? So I did a few years ago, back in 2015, we wrote this review where we looked at some of these key biophysical changes that occur as a function of age. And here I'm just showing you one of the plots, mainly in terms of morphology and mechanics. But what that showed to us is that there's quite a good whole where we can actually go forward and innovate and learn and gain new understandings. So when I think of aging in the sense of, when we look at each other, we can see things that are indicative of our age, right? So I think of it in terms of scales, where each of these scales, here I'm showing you 4, have an associated length and time component, right? So we can go from molecular to cellular to clinical to epidemiological, where these extremes, the epidemiological, we're thinking groups of people, and the molecular, we're thinking groups of genes. So today I'll drill in only on the cellular aspects of this work that my lab is doing, and really thinking of it through the concept that cell-based changes, or these cell phenotypes, are actually integrations of these molecular networks and patterns, right? So we have genetics, epigenetics, proteomics. These all integrate to give you these signals and cellular functions that we can see, right? And then once we have these dysfunctions arise, whether based on mutations or mislocalization of proteins or so forth, they lead to these dysfunctions in cells. And then these dysfunctional cells propagate throughout tissues and organs, and eventually manifesting as these age-associated conditions, right? So that is kind of one of the premise that my lab uses in terms of trying to show that looking at cells may offer this unique window, because it may allow us to predict some of these changes and predict some of these disease states before they happen, simply because changes in cells happen before some of these changes manifest at the clinical level. So we focus a lot on how cells move, their morphologies, and how they generate forces, which are all interconnected, right? So in order to move, morphologies change, the cells generate forces, their plasticity, and all these things change, with the overarching goal that we can associate some of these health outcomes and performances that are based on cell features with the chronological age to stratify or delineate healthy aging trajectories versus adverse aging trajectories, which will help us to inform in terms of disease. So when I started looking at this a few years ago now, we started off with a system of dermal fibroblasts. So we took these dermal fibroblasts, their primary dermal fibroblasts taken from the arm of individuals across ages, right? So we looked at from 2-year-olds to 96-year-olds, and pretty much asking what are some of these properties that change, both biophysical and biomolecular. And for the biophysical features, we looked a lot at motility, both single cell motility as well as scratch wound, more collective type of movements, assessing the traction strengths, meaning what kind of forces they're generating, as well as the deformability of the cytoplasm that we use based on microreology, right? And we also looked at morphology. So all these 5 classes of measurements altogether really gives us a biophysical picture, a biophysical snapshot of aging at these different ages in the dermal fibroblasts. And as what I'll show is that if we look primarily at the cell motility aspects of it, we can gain new insights that we may not have been so apparent looking at it in the more traditional or classical way. So we looked at these correlation maps, trying to understand which of these features associate strongly with age. And in these 2 classes, the biomolecular classes and the biophysical classes, we saw that many of the cell motility features associate strongly with age, with the strongest one being what we call the anisotropy, which we can think of as the directedness of the cell, tended to correlate very strongly. So if we use just the anisotropy, we can get a correlation of above 0.97 in terms of predicted versus chronological. And just to let you know that each one of these squares is actually a distribution of cells, right? So we're using actual single cell information to be able to reconstruct some of these correlation maps. And then we showed that we can use these features or pairs of these features to really predict the age of individuals and then stratify individuals based on whether expected aging or individuals who have a delayed aging pattern or an accelerated or premature aging pattern. So what I've done so far is essentially just laid the foundation to show that aging information is in fact encoded within these biophysical properties of cells, and we can use both snapshot and dynamic type features, meaning fixed and stayed morphology, as well as cell movements or cells tracked over hours, to really understand some of these changes that are occurring as a function of age. But one of the things we were still puzzled about was in regards to the question of how is this information encoded? So we know the information is there, but how is it encoded and propagated throughout the aging progression? So because cell motility and migration patterns and these are all spontaneous migration patterns, right? So they're not exposed to any gradients or anything. So I asked the question as to what can we learn by actually taking advantage of this single cell nature of the studies, right? And here I'm just showing a 3-year-old and cell movements for 8 hours, taking movies every 2 minutes in a 3-year-old and a 92-year-old. And when we look at these trends in terms of the anisotropy or the persistence time, you see this decrease in terms of these features as you increase age. So we can do a more thorough analysis, and here I'm using the anisotropic persistence random walk framework, which was proposed by Denny Wirtz's lab a few years ago, where it stems based on a framework that cells do not always follow random walk migration patterns, right? So from these framework, we can actually quantify many of the different features. We can ask questions about how do cells move in terms of displacement. Here we're looking at from blue to red, 2-year-old to 92, so young to old. And we can see this, this change in the displacement as you increase age, where cells displace less. We can look at the probability density functions and also see a similar result. So here you're just looking to see a transition from blue to red, which you generally see. We also looked at the autocorrelation function in terms of the velocities, where we're asking how coordinated these movements are. And we see that in young individuals, they seem to have a higher degree level of the autocorrelation function, which decreases as you increase age. And in addition, when we look at the angular velocity magnitude, we can also ask questions in terms of whether there's a spatial bias towards these movements, whether cells are moving very persistently or are changing directions very frequently. And we see going from young to old at an ellipsoidal profile to a circular profile, telling us that younger cells seem to be more biased in the direction, whereas as you increase age, these individual cells start having a similar likelihood of moving or sampling many different angles, right? So what we did further is really distill all this information into this heat map that I'm showing here, where we can use these 8 different features to define these motility trends as a function of age. I'm just showing you the 8 that are the most important 8 that we found. We've tested many other parameters and seeing the contributions, but with these 8, we see that we can pretty much recapitulate and explain this progression. So the takeaway message that we saw here is that generally there's a global decrease with age. Cells displace less, they change direction more frequently, and cells lose this polarized movement and they're less directed. But something that I thought about is in the sense that whenever we measure or we assess cell migration, we track these cells at the single cell level. Yet when we report them, we aggregate the data and take mean values and show averages, right? And looking through some of this data, it's apparent that some of the information that we can potentially gain by using the single cell nature of the data is lost when we aggregate it. So we started to go deeper into this along this concept to see what additional information could we get if we just keep everything at the single cell level and do a single cell based analysis type approach? So here I'm just showing you trajectories of all many single cells across different ages. And the slide before I showed you there's a decrease in motility as you increase age. And generally you can see the footprint is smaller in terms of going from a 2-year-old to a 92-year-old, right? So you can qualitatively see this. But when you start, so we plotted these 25 randomly selected cells within each of these categories, each of these different ages. And then you can see that it's not a uniform phenotype, right? It's a mixture, right? So in a 9-year-old, you can see cells that displace very far distances and also cells that don't displace much at all, right? And whereas if you go to an 85-year-old, you can also see some similar trends. However, it may be biased one side or the other, right? So that led us to hypothesize that this decrease in motility that we observe with age is not due to the decrease of motility of all cells, but it's rather based on this fractional redistribution across what we can think of as motility states, right? Or motility patterns. So we next tried to see, can we identify these motility patterns? And to do that, we again use these 8 features or 8 parameters that describe the motility patterns, which is the diffusivity in the primary axis, the total diffusivity, the mean square displacements at long and short time scales, the persistence time, this anisotropy, which you can think of as a spatial persistence, and also the persistence time and diffusivity in the non-primary axis, right? So we calculated all of these features for every single cell in our data set, which was about 860 cells. And then we use hierarchical clustering to see whether we can identify different groups of cells that may display different patterns, right? And what we identified was that there were about 8 different robust groups of cells that we observed, where, for instance, this group here in red, highlighted in red in the dendrogram, has very high normalized values of these features, right? So they high diffusivity, high spatial persistence, high temporal persistence. Whereas if we look at this blue group down here, a lot of it is blue in the sense that you have low levels of this diffusivity and persistence, and whether it be spatial or temporal. So to test to see how well this fits, we went back and selected all the cells within each of the categories and replotted the trajectories across each of these, where these 8 trajectories, we have, these are the cells that fit into P1, P3, P8. And when we look at them, we can see that they're actually distinct, right? You can see distinct homogeneous patterns as a function of these different groups. And you can start to identify cells that are very persistent and cells that move very far distances, as well as cells that don't move or are non-motile, right? And then if you look at, go back to these 8 parameters and you plot this radar plot that shows the normalized magnitude of these 8 features, you can see that they in fact are distinct and they have these different patterns. So they're defined. We've tested how robust these clusters are. They are very robust. So we have confidence in terms of going forward and assessing it in the context of aging. So just as a form of validation, we also plotted these 2-dimensional t-SNE plots where we try, and in t-SNE, we're just looking to see that these dots are clustered together. The length does not matter. The length scale has no meaning. So what we're able to see is that, yes, these groups that we saw in the unsupervised hierarchical clustering also panned out in terms of this t-SNE space. And interestingly, none of these groups are particularly defined based on a particular age, right? Which is something we saw from the qualitative data before, but quantitatively we can see that there's this intermittent distribution of all these different ages across these different motility patterns or motility clusters that we can go further and ask the representation. So since each of these dots represent an individual cell and each of them are colored based on the particular age, we asked whether how the distribution changed as a function of increasing age. So we plotted these bar plots here, where we go from young to older adults, where young individuals are considered less than 20, post-adolescent between 21 and 64, and older adults above the age of 65. And these cutoffs were decided based on literature values, right? So we looked at the aging literature and saw how these age groups were stratified. And interestingly, what we saw is that, you know, there's this changes in the abundance of these cells as you change with age, where if you take the average of the first 4 and the last 4, and we're taking the average because if you remember the dendrogram, there was one main bifurcation which led into 4 groups and 4 groups. We saw this interesting pattern, right, where from patterns 1 through 4, it was high going to low, high going to low for most of the young individuals. However, when you looked at the older adults, you saw the opposite trend, right, in the sense of low going to high, low going to high. And these post-adolescent adults had more or less, the line was almost flat, right, in the sense of the distribution of cells in 1 through 4 and 5 to 8 were comparable, right? Starting to tell us some things about heterogeneity, that there may be increased heterogeneity within this post-adolescent older adult population. So one of the other things which, you know, it was just a curiosity of mine that I think you can gain from this type of analysis is, can you assess these age-dependent motility trends in terms of asking questions such as, we saw that displacement decreases with increasing age, and also persistence decreases with increasing age. Which one changes first, right? Is there an order in terms of how these patterns change? So we can use these spatial clusters to really try to define a likely progression order in terms of these events. So I'm just going to show you the results, but we essentially use these correlation analysis where we use the cross correlations across clusters and the correlations with age, and we can map out the progression order, where this map here, the size of the node represents the number of cells in the cluster, and the length of the link tells you the cross correlation, right? So as a function of age, the order seems to go from P1 to 3 to 2 to 4 to 6, where you can see these cells are, you know, displacing very far distances, not necessarily very persistent, and then you get more persistent, persistent, right? So what this data shows you, and I just showed you qualitatively here, but what we can see is that the age trend seems to lose displacement before it loses persistence, right? So these are some of the analysis that we can push forward with this. So these spatial patterns allowed us to really understand that, you know, there's this redistribution of the abundances as you increase age. And it's not just a decrease, right? So we can understand a deeper information about it. But we also had questions about the activity in regards to the burstiness in terms of the dynamics, right? So to assess the activity or to quantify the activity of each cell, we took these 2-dimensional trajectories and decomposed it into a 1-dimensional displacement, where now this, the y-axis here is just a z-score of the displacement. This is the elapsed time. And then we can actually quantify these patterns in terms of the burstiness of the movement to really understand something deeper about the movement patterns of these cells. So we went ahead, did this for every cell, and then took all of the 860 cells, reclustered them, identified these 4 activity patterns, which, you know, these are just showing 5 examples of just randomly selected cells within it. And you can see that AC1 is different from activity cluster 2, which is different from 3 and 4, right? But interestingly, when we actually plotted the trajectories, we didn't see distinct patterns as we saw with the spatial ones, right? Which starts to tell us that, you know, cells that displace far or cells that displace very little could show similar responses in terms of their activity and the burstiness of their movement. So we tried to simplify this and still see how best we can quantify this activity. And we use this point process analysis, which is based on some signaling type analysis, where we can define trains and lags, right? Where trains are defined as the number of points above a certain threshold that we determine based on the data, and the lags are defined as this the points that are below that particular threshold. So we went back, did that analysis, and we quantified these average train lengths and the coefficient of variance of these train lengths, and we're able to essentially describe the patterns with age as a function of these activity clusters. For instance, where we saw AC activity cluster 1 was largely depleted in older adults, and activity cluster 2 was largely where an activity cluster 1 was enriched within these young individuals. So because we can now stratify or classify each of these cells within each of these spatial clusters as well as the activity clusters, we went back and co-classified everything as a function of age, young, post-adolescent adults, and older adults. And also we're able to quantify the statistics in the sense of enriched versus depleted based on these spatial and activity clusters. And there's many take-home messages that we can take from this, but one of the key ones are really dealing with the concept of Shannon entropy and the heterogeneity, right? Where we're seeing that there seems to be this significant decrease in the degree of heterogeneity
Is it just me, or did you disappear?
I think we lost him.
Okay. My internet connection was also being a little bit unstable, so I thought it might have just been on my
He's come
side.
back. He's coming back.
He's come back. All right. Let's just give him a minute to get things set up. Hey, Jude, can you hear us okay? Let's see. Jude, if you're able to talk but not share your screen, we could also jump into questions. If it's a screen share, that's a problem.
His microphone doesn't even seem to be connected on the participants list. So I think if you can hear us, it's probably best if he leaves the Zoom and then re-enters, I guess. But he was having problems before as well.
I know, but it worked perfectly for a bit. Let's just see if we can give
no,
him
he's gone from the participants list, so I think he's going to reconnect in a second.
Okay, sounds good. It's funny, they always tell you that you should connect from work instead of from home, but I feel like the only problems that people have is when they're connecting from the lab.
He's back.
Hey, Jude.
I seem to have cut off. I was like talking the whole way and then just realized everything died off.
That's my worst fear. All right. Yeah, well, we're coming towards the end, so probably you were sort of close to wrapping up, I think.
Yeah.
Okay.
Yeah, I can just go back to like my last few slides. So yeah, so the main takeaway from some of this is that we can co-classify cells based on spatial patterns and these activity patterns that we can we can do the statistics in terms of to identify which ones are enriched or depleted. But I think one of the key takeaways from this is that we're seeing this change in heterogeneity and entropy as we go from these post-adolescent adults to older adults. So the main take home is that although we are able to see this global decrease in age-associated trends in terms of cell movements, cellular heterogeneity and potential state transitions seem to drive some of these observed phenotypes, and that the aging information seems to be encoded within the diversity and the heterogeneity of these cellular phenotypes. And I'll just give you one slight application in the sense of, we've been able to define some of these different clusters and patterns. One question we had is can we apply this same framework to classify cells that we didn't use initially in this classification? And so we took additional fibroblasts from, these are mouse embryonic fibroblasts, and then we tried to use this, build a classification system using machine learning approaches, where we show that with a 98% accuracy, we can classify fibroblasts, and these are, MEFs, wild type, and then you knock out protein A, and then you're able to see these changes in the distribution. So we're building on this, and we would really like to build off a platform where we could identify patterns of movement across condition, regardless of cell type treatment, and see whether we can identify these global, if you will, patterns of movement. And we think that we can. We've started this, and I'll give you like a little sneak peek in the sense that we've developed a way to take, and we have data for about 25,000 single cell trajectories that cover about 400 conditions. We developed new approaches for data harmonization, where we brought everything to a total duration of 8 hours and interval of 5 minutes, and then we can do our classification and predicting the state. And what we've seen is that with these 25,000 trajectories, we can classify them, we can get these different distinct patterns of movement. These clusters could also be substratified into different groups that could help us understand better the patterns. And then, we can go back and quantify all these different trajectories in the different groups. But I think the main take home I wanted to show was that in the sense of we can take examples of cell movements, right? So here I'm showing you T cells that are embedded within collagen gels, collagen 1 gels of different concentration. We know that there, you see this biphasic type effect. You hit the biphasic in terms of persistence time, in the primary axis, non-primary axis, the total diffusivity. I can use either 1 milligram per ml or 4 milligrams per ml in the sense that they'll give me comparable persistence time or anisotropy, or they may be different in diffusivity. But what we see is that if we can use our co-classification system in these groups, we see that it's more complex than just a decrease or comparable levels of persistence time and anisotropy, but it's actually still, you can see this redistribution as you change, where, for instance, as in conditions 2 milligram and 3 milligram, you see the emergence of this cluster here, which is not as apparent in the other ones. So 1 milligram per ml and 4 milligrams per ml, although they may have similar persistence time, are very different in their movement. And then we've shown that we can co-classify all these cells that it had never seen using a SVM classifier, and we get here I'm showing you a 95-5 split, but we can we've done it for 20/80, 90/10, and so forth, and we still get a high level of accuracy. And with that, I would like to thank you, the audience, for listening, and all the persons who contributed to this work, particularly the work I showed today from my postdoc, Deben El-Meiti, and a close collaborator of mine, Nawal Zamponi. And also a quick plug, I have a postdoc position opening, so if anybody's interested, definitely feel free to reach out. And thank you.
Wonderful. Thank you so much. And hopefully somebody in this audience will be getting in touch with you about that position. So let's start with a question from Joshua Doloff.
Hey, Jude, I love your work, obviously. It's cool stuff. It's fun seeing your recent paper updates too. I'm curious explicitly about the cluster heterogeneity you're seeing within each cluster. And so there are a couple possibilities, obviously, maybe the way you're getting cells, you're collecting multiple cell types, or there's some contaminants, or it's perhaps plasticity or a state of differentiation. When you take those cells with those markers, you have different subclasses of cells. And then beyond that, there's also the bigger concepts of you're getting cells from all these different patients at different ages. Are there bigger implications in terms of differences in genetics or demographic background and body chemistry? Can you speak to these things? Yeah,
so those are all fantastic questions. I think you hit on like a key a couple key points that the direction that we're pushing towards. So we are trying to incorporate some of the molecular data and the patient type, like the clinical data with these cell motility type patterns to see how we can further stratify that. In regards to the heterogeneity within the cluster, I think, the more data we have, I would expect like those to kind of, to separate and to become cleaner. But you also touch on a good point, which is this concept of state transitions, right? Where you could potentially have the same way I showed a progression order, where you can think of, losing displacement versus losing persistence. You could think of a progression order in terms of states, right? Even within the same individual. So those are things we're looking at. Yeah, I'm super excited about.
Fantastic, thanks. We also have a question from Fernando Valencia.
Oh, hello. Thanks for the great talk. I just had a quick question with regards to, like, if you look at commonly used immortalized cell lines to see kind of where they fall with age. I also think like using like a common immortalized cell line that has been kind of studied more with regards to proteomics might be kind of interesting to give you kind of like insights into maybe differences within the proteome itself.
Right. So we haven't looked at that, but the goal is that we're trying to put together. So the last part I showed you very briefly, we're trying to put together that method and a manuscript out soon on that, where the hope is that anybody could take any of their data and push it within our framework that you can co-classify your cells within that. So those questions open up in the sense that you can start asking, how are these aging trends similar to some of these perturbations or cell types or cell states within some of these. However, I do think that like these aging patterns may have some cell type specificity in the sense that I'm not sure exactly how, if you take any immortalized cell line and you compare it with fibroblasts or so, how that will change. But that's an interesting question, something we could look at.
Great. We have one more raised hand, but I'll just ask, we have a couple questions on YouTube that are both asking about stiffness and the role of age-related changes in stiffness and how that might be playing into some of your results.
Yeah, so definitely we know that stiffness plays a part, and a lot of studies in terms of these changes with aging show that the extracellular matrix and tissue stiffness actually change, right? So all the work we've done here and the ones we focused on is looking at more cell intrinsic changes, right? So we've seen that cells actually are capable of, they have some sort of memory, if you could call it that if you take them out of context, they can still recapitulate certain trends that we see. So these trends that we're seeing in regards to changing in age may most likely have some semblance of a memory from the stiffness that they came from. However, we have not tested that yet.
Okay, great. And then just quickly, and then we'll move on to Shashank, running a little tight on time. Claudia Vasquez, if you want to ask your question.
Yeah, thanks so much for an awesome talk. I was curious, you were saying something about just now in the previous question about how different cells have different kind of dependencies on aging, kind of in the more like cell culture world. But I was curious if you had any thoughts or maybe like ideas on which cell types, like do our immune cells or dermal cells more sensitive to aging, or is that like, yeah, I would be curious to know which cell should I be protecting most?
Right. So we're not sure on that yet. We're in the process of doing a bunch of these. We're actually setting up a platform to be able to do it with immune cells. I think immune cells is an interesting one to look at. One, you can get them easily from blood, and they sample through multiple microenvironments in your body. So they may have some sort of ensemble age imprint in these cells. So those are particularly interested. Whether one cell type may be predicted versus aging or not, we don't know yet.
Thank you.
Thanks.
All right, great. Thank you so much, Jude. That was really interesting. I also have a couple questions, but maybe we can follow up afterwards. And I see there's a question in the chat from Raphael, but maybe you can answer that offline. All right, so